Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Here we solve questions from various MSc entrance examination. So today's question is from TIFR GS examination in biology. Year is 2017. What is the correct order of action of these enzymes during DNA replication? Helicase, primase and polymerase. The three enzymes are given and different orders are given in each option. So we have to find out the correct order of these events. Let's have a look at each and every enzyme so that we can find the correct order. So let's understand what these three enzymes are: helicase, primase and DNA polymerase. You know that DNA is a double-stranded structure. The two strands are wound around each other. So for the starting of replication, there must be an unwinding of DNA strands. So for this unwinding, this helicase is there. During the starting of replication, the first enzyme which acts upon is a helicase. It breaks down the hydrogen bond between the bases. A and T is having double bond, whereas G and C are having triple bonds. So these bonds are broken down using helicase and the two strands of DNA get separated so that the replication can be done. Right? The helicase also works during a DNA repair mechanism during DNA recombination. Also it is important when the conjugation has to be occur. So wherever the bond breakage, hydrogen bond breakage during DNA two strands is required, helicase is there. The second one is a primase. This is the enzyme which synthesizes the RNA primer. For the synthesis of DNA, there is a requirement of RNA primer which is fulfilled by this enzyme. The name is primase. This synthesizes RNA primer having around 15 nucleotide sequence and that is attached to the leading strand. It can work in a 5' to 3' direction. And this primer is very essential for the starting of the DNA replication. The third one is a DNA polymerase. This is the main enzyme which is required for the DNA replication. This uses a DNA template and using that it replicates the DNA strand. So it is also known as a DNA dependent DNA polymerase. Here in bacteria there are three types of DNA polymerase, 1, 2 and 3. So the main replicative enzyme is DNA polymerase 3. Whereas this DNA polymerase 1 functions in a DNA repair and gap filling. The DNA polymerase 2 functions in a DNA repair. So in this way it has a three types. When we think about eukaryotes, the DNA polymerase is of type alpha, beta, gamma, delta, epsilon and kappa. So it is having different functions. Delta and epsilon are the DNA polymerases which are used for the DNA synthesis. Leading strand synthesis is done by DNA polymerase epsilon, whereas lagging strand synthesis is done by the DNA polymerase delta. This DNA polymerase alpha, this is required for priming during replication. The second, that is DNA polymerase beta, this is involved in a base excision repair system. Secondly, DNA polymerase gamma is involved in a mitochondrial DNA replication. Mitochondria is having its own genome. So it is having some of the enzymes which are required for its replication. In that sense, DNA polymerase gamma is there for the mitochondrial DNA replication. Then this DNA polymerase kappa is for cohesion. So in this way, it is having different functions, and these many types of DNA polymerase we find. So the correct answer for this question is option A. Helicase, primase and polymerase. Helicase is used for the unwinding of the DNA strands. Then primase comes and binds the primer, RNA primer, to the DNA. Whereas polymerase is there for the DNA replication. Let's have a question for my next video. The question is from TIFR GS Biology Entrance Examination 2018. The question is regarding the evolutionary sequence. The organelles are given and it is asked that which organelle has arose first during the evolution. We'll solve this question in my next video. Till then, don't forget to like, share and subscribe my channel, and also to hit the bell icon. Thanks for watching.